Hello and welcome to the WeXtree Online Academy. My name is Chris and I'll be your tutor for today. In our previous video we looked at questions 11 through 15 and today we'll look at questions 16 through 20 of the Cambridge As Biology Winter Paper 1-1 2019, course code 9700. Okay, question 16. Cell surface receptors used in cell signaling are usually proteins that extend from one side of the membrane to the other side of the membrane. What is the role of the extracellular part of the protein? Well, this means it's sticking out on the outside of the cell. And cells recognize each other by bumping into each other and bonding with that particular molecule, whether it's an antigen on another cell or a protein on another cell, or it's a substance that's in the blood and it recognizes it as and it wants to draw it into the body of the cell, by whether or not it's pinocytosis or endocytosis, where it encapsulates something outside. Okay, but what happens is it acts as a binding site to a signaling molecule. Okay, that signaling molecule could be, let's say, on the cell or a substance, it doesn't matter, but it's to recognize and attach to it. It doesn't function as an enzyme. It does not change the solubility of the substance, and it does not transmit signals from the target cell. Okay, it's not a neuron, it's not transmitting impulses or any other chemical messengers. It's just to bind to whatever it's come in contact with. Okay, so option A is the answer for question 16. Question 17. Which substances can pass directly through the cell surface membranes and do not use carrier protein or channel proteins? Okay, so we have potassium and chloride ions, carbon dioxide, and glucose molecules. Okay, so remember that in order to pass through the cell surface membrane, this has to be a process of simple diffusion. Okay, whereas carrier proteins or channel proteins, there'll be active transport or facilitated diffusion. Okay, so mineral ions generally are moved back to transport and therefore would need a carrier protein, or in the case of specific ion channels, they'll move along the channel proteins into the cell accordingly. Okay, so this one is not going to, this uses a channel protein or a carrier protein, whereas carbon dioxide does not need a channel protein or carrier protein. Okay, so this one is a potential option here, and glucose can move into cells by either facilitated diffusion or by active, like primary or secondary active transport. However, they do require carrier proteins to be able to do this. So the answer to this one is simply only carbon dioxide can pass directly through the cell surface membrane by simple diffusion, which gives us option 2 only. Okay, eliminating A, B, and C. Question 18. A student put a layer of plant epidermal cells on a microscope slide. The student put a drop of potassium nitrate solution on the layer of cells and observed that the cell surface membrane of many cells had separated from the cell wall. The cytoplasm contents had shrunk. What explains these observations? So looking at the options here, we know that the description correctly defined as plasmolysis, it looks like the cell has become plasmolized. And as a result, this means that water has left the plant cell by osmosis, leaving it all shriveled inside. Cell membrane is detached from the cell wall, and the actual cytoplasm has shrunk in relative size. Now, this means that it's gone from cells to solution. So from this, we can eliminate options C and D from the choices here. Now, the confusing part for some people is that there's negative numbers here. So we have the starting water potential of cells and the starting solution or water potential of the solution. Now, technically, minus 100 is a higher water potential than minus 500. Okay, the reason is negative numbers, that might confuse you. And if we understand that, then we know that osmosis goes from high water potential to low water potential, okay, which gives us option A as the correct answer. You know, this one describes movement from low to high water potential, and that's why option B is incorrect, leaving A as the option. So, question 19. The enzyme telomerase prevents the loss of telomeres after many mitotic cycles. Which cell transcribe a high concentration of telomerase? Okay, so you've got neutrophils, mature red blood cells, and activated T lymphocytes. Okay, so well, we can eliminate option 2 straight away. That does not contain a nucleus and therefore not carrying out any cell division. The neutrophils are a very highly specialized cell. These are created in the bone marrow And once they've been released into the circulatory system, when they find a place where they need to migrate into the body tissues, they'll then die pretty soon after that, about a week after they've migrated across the capillary wall and into the body tissues. They do not carry out mitosis themselves. They'll have to be replaced through mitosis from the stem cells, okay? However, activated T memory cells do need the capacity of their cell division, and the high concentration of telomerase prevents the breakdown of that end bit of the telomeres, which contains important genetic code about any particular antibodies that need to be made, possibly, or any other important information held within the DNA. If this was not there, they would lose chunks of code every time the mitosis would take place. Okay, so that leaves just option 3 is the correct answer. And the only one here is D. So option D is the correct answer, eliminating A, B, and C from your choices. At which stage of mitosis do these events take place? Well, if you remember in mitosis, you've got PMAT. Okay, so the key understanding here is understanding what these things are. So P is prophase, metaphase, anaphase, and telophase. This takes place in this particular order. In prophase, this is where the chromosomes condense and clearly become visible under the light microscope. You can see this as you're part of your core practical. The nuclear envelope is broken down, but they are visible at this point in time. During metaphase, you can see the chromosomes have lined up along the equator of the cell, and spindle fibers have formed attaching to the centromeres of those chromosomes that are lined up. Okay. During anaphase, this is where the spindle fibers contract, and this will pull apart the chromosomes, breaking them apart at the centromere. And in telose phase, the nuclear membrane has started to reform, and then the cell is getting ready to divide and then you have cytokinesis. Okay, so here you have centromeres separate. Well, we've established that only anaphase is the phase where the chromosomes actually separate, all right? So your choice now is between interphase and prophase, all right? Now, outside of these 4 processes here, interphase is a very large section, okay? You've got G1, S, and G2, okay? So in G1, it's still growing, and normal functions are going in place. So the cell's just getting bigger. In S phase, you've got the synthesizing of new DNA and getting ready to move into G2. And G2 is where you still have normal cell functions, but it's getting ready to divide. So the cell's very big by this point. It's replicated enough DNA to undergo mitosis, and therefore it's ready to undergo mitotic phase. Okay, so during chromosomes become shorter and thicker, well, this has nothing to do with interphase, because here are the 3 parts of interphase we just discussed. This eliminates option A, leaving option B as the correct answer. Thank you for watching our video today. Please register on the website for a free demo, and I look forward to seeing you in our next video.